All right, well, welcome everybody to the first epic of the spring here. I'm Dr. Johan. I teach biochemistry and general chemistry here. I'm also an avid home brewer. Some of you may have gone to my first home brewing talk, and so I talked about various things related to general chemistry at that talk. This one, I wanted to pick up other things from general chemistry, but also I wanted to talk about organic chemistry in terms of wine. And I'll tell you, when I was putting this together, I discovered that really a lot of the things I thought would be organic chemistry in wine were actually microbiological. So they were bacteria doing things in wine. So it's not going to be as much organic chemistry as I had hoped. I know, and there'll be some tangents. And there'll be some places where I need some help with mechanisms. So maybe the organic chemists out in the audience will be able to help me. So to start off, I was going to talk about supersaturation, but the demo I wanted to use for supersaturation was missing from the stock room. So I've created one here, but I think it probably needs to cool off. All right, so I'm going to let it cool off over the talk, and we'll come back to the first slide. So in terms of brewing, why don't I'll quick talk about in terms of brewing what kind of the process is, right? I forgot to do an overall process for brewing, but what you do is you take, in terms of wine, this thing right here, came to my last talk, you saw me put one together. This is like 2 quart, this is, well, actually quarts because it's a gallon. This is like close to a gallon or 3/4 of a gallon of the cheapest grape juice I could get at Kroger, 2 cups of sugar, fast rise bread yeast. This is my cheap and easy wine. And then I actually add a little bit of ammonium diammonium phosphate to it. So diammonium phosphate is going to look like this. Anybody remember your charge on phosphate? Polyatomic ions? -3, actually. You're guessing 2 negative because there's an error here. All right, so in terms of phosphate, what you do is phosphate is going to be -3, right? So for this to actually work, you're always going to have your charges balanced with these sorts of things. There's going to have to be one of those phosphates is going to, or one of the oxygens on the phosphate is going to have a hydrogen on it. Okay, so this is what diammonium phosphate looks like. Why would I add that to wine? Why would I add that to my grape juice? Anybody have any ideas? If you're growing plants, what do you do? What's one of the things you do if you're growing plants in your garden? You water them. That's good. Watering's good. What if you don't have good soil? You fertilize them. What goes in your fertilizer? You guys know what's in that fertilizer? There's not necessarily sugars, but plants need sugar. Plants would love sugars, but I don't know if you can take them up or not. What do you think? Nitrogen, right. Do we have nitrogen over here? Yeah, we have nitrogen. So plants can't make their own nitrogen. Some plants actually have a symbiotic relationship with bacteria where they can do that. So nitrogen in the air looks like this, right? List out structure for nitrogen in the air is that. All right, so that's nitrogen in the air. What do you think about the bonding between those two? Weak, strong? Strong. It's a triple bond. It's really strong. So this is super hard to break this. So plants on their own, and most plants don't have this symbiotic relationship with bacteria. It's like beans and clover and things like that have a symbiotic relationship with bacteria. But they don't have the ability to break these two things apart and make the nitrogen useful for them. So what you can do for those plants is you could fertilize with ammonia. This would be ammonium. This is what ammonia would look like if it was in water. Okay, so you give them nitrogen that way. And so what we're doing here is one of the things we're doing here is we're giving the yeast some ammonia or some nitrogen, a nitrogen source there. Okay, I've got the grape juice. I try to get as much of the sludge from the bottom in there for extra food for the yeast to grow. But what I found is when I started brewing, I got really, so you start brewing, you're really excited. You set up a bunch of things, but you have to wait, right? So you don't find out whether they turn out or not. So when I started brewing, I made, my very first thing was I made an apple cider from straight, or a hard cider from straight apple juice, right? Made it, turned out really quite good. I was surprised it was good as it was, because it was, of course, I'm cheap, so it was the cheapest apple juice I could find at the store. But I made this stuff and it was good. So I was like, okay, I'm going to make it again, but I want to have more alcohol. So I added sugar, right? You added more stuff for the yeast to turn into alcohol. Okay. So if we think about what yeast do, you're going to give them sugar. I'm going to put up glucose. I'm giving them sucrose. But what they're going to do is they're going to take this, right? They're going to use that to grow while they still have oxygen. But when they run out of oxygen, they're going to go ahead and turn that into ethanol. I'm not going to balance this. Okay, this is what they do when they run out of oxygen. So if you give them more sugar, they're going to make more alcohol for you. So I thought, okay, great. I'm going to super alcoholic apple cider. And it was awful. It had this sulfury taste to it. My wife, who's very tolerant with all things I brew and drinks like all of them almost, just couldn't stand it. Okay, this was kind of during COVID. So I actually got COVID. And I was like, okay, well, maybe I can stand drinking these now because I can't taste anything. And it was still coming through. I actually drank it anyway, but I'm pretty tolerant. But I couldn't taste anything. I was still getting stuff through, so I guess I could taste a little bit of something. Anyway, so really bad stuff. And what I found out, I was kind of looking around doing some research, and what some people said is, hey, if your yeast they run out of stuff, if they run out of ammonia and they run out of phosphate, they start doing weird reactions to try and compensate with that. All right. So the point of this is to give them a little bit of extra nitrogen, a little bit extra phosphate, so they don't get so they're not really stressed, and they make a lot of things that you really don't want to taste in your brews. Okay. Particularly things like sulfur compounds you don't want. Okay. Questions on that? Probably not. Okay. So yeah, I add that as well when I do this. All right, so you've got this process. You throw the wine in. I've got my bread yeast in there. The yeast are going to go and they're going to make ethanol. Okay, but you guys have probably heard about aging wine, right? You want to age wine. If you age cider, actually, it gets better over time as well. And then it kind of probably gets worse, I would say. But what you have is if you take cider that you've just made, so apple cider, hard cider that you've just made, what it has in it is it has malic acid in it. Let me see, do I have the structure? I don't have the, no, I do have the structures. Okay, so we'll do that. So it has malic acid in it. And what that is that's got the taste of green apples, which, isn't a bad taste, but when it's really strong, it's not such a great taste, right? You're thinking about green apples from the store and they're still sweet. I'm thinking about green apples like off the tree that are like this big and it's a really strong, sharp taste. It's not quite good. So if you let that thing age, what happens is the bacteria there are bacteria that grow in there, right? You've got yeast that you use to make your ethanol, but while you didn't add bacteria, there were some bacteria in there because you weren't careful about making everything sterile, right? So I try to sterilize things, but really, I used to try really hard to sterilize things. Now I just clean things, okay? And I know I'm going to get a little bit of something, and it's okay. But what happens is this is a lactic acid bacteria. And so your yeast come up, they make all the alcohol, and then they've eaten all the sugar at this point in your solution. Okay, they eat all the sugar, and so now they're kind of trying to hang out. Maybe they're eating a little bit more of the sugar, but really there's a lot of alcohol in there. They're having trouble getting along, and so your yeast population is going to decline here. And after some amount of time, you've got some bacteria you got in there, The wine industry, they actually add this specifically. Okay, they know what bacteria they want to make this happen. They add them specifically. They're timing it. For me, I just let stuff sit in my basement for a long time. Okay, and then I'm going to get one of these. I don't know that it's exactly the species, this Ooni that we've got over here, but they're going to start doing malic acid to lactic acid fermentation. All right. So what I found with my ciders, I actually have some ciders that have sat in my basement for over a year. I think I probably got some things in them 2 years down there. And they go from having this very sharp taste to what I would say is really flat. Like there's something in the middle is where you want to hit it, right? You do want it to apples to have a little bit of that sharp taste, but you don't want, but you don't want a total absence of it. And so this is the reaction we're talking about here. So this is malic acid. Both folks that are in biochemistry will recognize the structure. And these bacteria are going to take that malic acid and they're going to decarboxylate it. And they're going to make lactic acid. Okay, and this is the lactic acid. It's the same lactic acid that you make in your muscles when you're working out. Okay, and you're not. And what happens in your muscles when this is going on is you're not getting enough oxygen in. So you're working out really hard, your body's not bringing enough oxygen to completely oxidize all the fuel, and so you have to make some lactic acid. And I'm not going to talk about why that is here, but if you take biochemistry, we'll talk about it. So I was looking at this, and there's an enzyme, malolactic enzyme. I'm a biochemist, and I was interested in that enzyme since we were doing this. So this is, preparing this talk was kind of me wandering around looking at various topics. And I found a paper from, it's not here, but it was like 2012 where they actually isolated this enzyme and they overexpressed it in bacteria and characterized it from this is the main bacteria that actually does this for wine. And my first thought was, oh, it happened in 2012. Nobody had done it before. I could have done that. Okay. It is actually something that in my old wet lab research group, we could have done in the lab. Okay, so these are the sorts of things you can do in research. And what they did is they characterized it. So they characterized the activity of the enzyme versus what the pH of the environment is. And so you see here the enzyme is most active at, say, a pH of 6, 6.5. What do you think about that? Does that make sense based on kind of where we see this bacteria, what it does? Where are we seeing this bacteria? Okay, so we do have acids around, right? In the wine. Is wine, when you think about wine, are wines like kind of acidic? Are they kind of a little acidic? Are they kind of a little basic? More acidic, right? Okay. You guys know what bases taste like? What do bases taste like? Bitter. You guys know bitter? Okay, so bases are bitter. All right, so the reason why a lot of pharmaceuticals taste really bad is because they have amines on them and they're basic. Okay, so bases are bitter. I'm trying to think of good bitter foods these days. You know, broccoli's a little bitter. Brussels sprouts used to be really bitter, but they changed the cultivar. Now they're not quite so bitter. I got a suggestion, Dr. Dan. Bitter foods. Greens, really dark greens can be bitter, so just in terms of that taste. So not surprising, right? Slightly acidic. pH 7 is going to be neutral, slightly acidic. That's where the enzyme works best. All right, great. Okay, that makes sense to me. But then I was surprised by this. Here we have the temperature curve of where it's most active, right? So this is Celsius down here. Can you guys see this? This is 20, this is 60, this is like 45 to 50 degrees Celsius. What temperature is it in the room right now? In Celsius, roughly. 20-ish, right? It's about 20 degrees in here. 21 degrees because it's hot. Okay, but 20 degrees or something like that. So this stuff sits in my basement, right? Or it sits in a winery. And when you picture wineries, do you picture them being at, I mean, you can't even live at this, right? I don't know what that is in Fahrenheit, but it's well over 100 degrees. Okay. What was that? They're not really warm. They try to keep things cool, right? They try and keep wine cool and things like that while you have this going. So I was really surprised that this was most active here. Now, part of this, my research is I work on, my research now is computational, right? So I go and I take proteins and I model proteins binding to things on the computer. And I thought, wow, well, okay, so these guys just made this. So my other thought was, I bet you there's not a crystal structure yet then. If nobody's overexpressed it before 2012, there's not a crystal structure. And what a crystal structure is a crystal structure is a picture of a molecule. So how do they get those crystal structures? All right, if I want to get a crystal structure of a molecule, the way I do it is I actually do get a crystal. So if it's a protein, I grow a ton of the protein. I make a ton of the protein on bacteria. I purify it, the painstaking process, and then I do a bunch of random stuff to it, maybe slightly guided, and I try to get it to crystallize out. How many of you have done crystallization in any of your labs? Was it easy? It might have been. We would have given you an easy crystallization, but you might have had problems with it, right? You're trying to get this stuff to crystallize out. This is really hard, okay? So what people will do is you'll have your protein crystal and you'll have like a bunch of a robot and a bunch of 96 well plates, which are like little beakers, 96 little beakers together. And you will try a bunch of different types of solutions, hoping you find a magic one that will cause you to make protein crystals. All right, and so often this is the hardest part of this work if you're doing it for your graduate work. So you get this tiny little crystal, and then you put it on the end of a stick, and then you shoot X-rays at it, okay? And what happens is the X-rays go in where you've got atoms. So imagine, so here's an atom, here's an atom, here's an atom. The X-rays come in, and where there aren't atoms, so there's a hole where there are no atoms, right? Where there aren't atoms, they go through and they diffract, like a diffraction grating, or if you've ever seen like, diffraction grating is my best thing. You ever have guys know what CDs are? Okay, just checking. The things keep changing. So CD, you take it on the light, right? You take it, you see the different colors, right? It's taking the different colors and it's diffracting them at different directions. So, but we're going to shoot this through the crystal, and the X-rays are going to be deflected, and they get deflected in a way by where there are holes. Okay, and you'll get this pattern of spots behind your crystal. And then what you can do, now it's all done by computer programs, you used to do this by hand, that you can, from how the spots go, you can infer back where there were holes and where there were, these are electrons, where there were electrons around the atoms. Okay, so now I know where there were electrons around the atoms, and I know what the pieces of my protein are, and I can then try to fit, if I have a piece of a protein that looks like this, right? This is a heme with an iron in the middle. If I have something like that, does that fit my electron density maps? The computer tries to overlay things for you, and it does that for an entire protein. So it's just a little piece, you do it for an entire protein, and you get these beautiful pictures we have where we know where the atoms are, okay? So that's how we get these pictures that you may have seen a bunch of these before, you may not, if you see them in the future, that's how they're generated. So, turns out, no crystal structure, right? But I'm in luck that people invented artificial intelligence, right? Not invented, but there's been a lot of work on machine learning that's really taken off in the last decade here. And so there's this problem in protein chemistry. There was this problem. It's still a problem, but there was this problem where people were trying to take the amino acid sequence of a protein. So you could imagine, I'm just going to write letters. I'm not going to do the structure much here, but we're going to take an amino acid sequence. So let's say I know I have an alanine, a glycine, a lysine, alanine, etc. Okay. So DNA sequencing is great. You can do DNA sequencing fast on things. So you can see we've sequenced the DNA of so many organisms at this point. And so we can take that DNA and we can translate that sequence and get a protein sequence out of it. All All right, so we know what the sequence, the linear sequence is of our amino acids. And then the question is, if I know what that is, can I figure out what the shape of my protein is? And this is something that people have been working on for the last 4 decades. Okay, and up until about 5 years ago, we figured we were still 3 or 4 decades off of getting it. People were trying to make computer programs that use where are the hydrogen bonds, where are the pluses and minuses, where are the London forces, and tried to put that all together to get shapes. Okay. And what these guys did, these guys got the Nobel Prize for this. The group who did it is much bigger, so they must have been just leading the group. These guys were leading that group to design this program called AlphaFold. These guys are at Google Mine, which is it's part of Google, and it's in England. Okay. So what they did was we got 3 or 4 decades off of solving this, and they largely solved it using AI. Okay, and so it's like your Chat GPT. We don't really know how it works, but it works. It's really doing it by pattern matching the things that are out there. Okay, more than by rules, by this is hydrogen bonding to this, or we have a plus and a minus next to each other, those sorts of things. Okay, so they've done that. So they could take every possible gene we know and they could come up with a structure for it. And so I was able to grab the structure for this enzyme off of that. And I look at it, it's kind of big. Maybe I would have had a little trouble doing it in my lab. Might have been still how to do this is not tough. And in fact, people who are in biochemistry right now are doing this in our biochemistry class, not in this enzyme. Okay, but they're doing it to different enzymes. They're taking proteins, they're putting them in E. coli, and they're what we call as a bacteria. They're putting in bacteria, and they're making lots of them. Anyway, cool structure. I have no idea where the active site is or where things bind to it. It could be fun to play around with. And if I have time, maybe I'll do research on that at some point. Anyway. But wanted to talk to you about this. So this is really this AI stuff. You guys are probably used to it or whatever, but really this is a sea change for this. When this first came out, I didn't believe it. Okay. All right, let's go on, and I'm going to talk about enzymatic browning in wine. Okay, so one of the things that happens in wine is when they're making it right at the beginning. Okay, you take grapes and you smush them up, right? And what happens is you can end up with some very brown colors. If you're making a white wine, you care about that, right? You want it to be clear. You don't want to have a lot of dark color in it. If you're making a red wine, you're not so concerned because people aren't going to see it very well. But why does this happen? This is the same browning that happens on your apples. It's the same reactions. Okay? And so what you have is to get that browning, you need 3 things. Go ahead and do this over here. And the 3 things are up here, but let's say this is my apple, and my apple is made up of cells, right? And so inside those cells, what I have is these phenols. I'll just do those as pH. Okay, I've got phenols. That's a phenol. I'll do a PHOH like that. Okay, I've got phenols in the cells. And then also in those cells, I have an enzyme that's called polyphenol oxidase. So we'll do that as PPO in those cells. Okay? Then outside the apple, I have oxygen. And I need all 3 of those things to come together to get the browning reaction. So what happens is when I cut an apple, and here they've clearly treated part of that apple, but when I cut an apple open, I am breaking these cells, okay? I break these cells open, I break open this compartment, and I let the enzyme mix with the phenols and with oxygen when I break open that apple. And what that'll cause is now my polyphenol oxidase can start putting extra hydroxyl groups onto this, and that it can oxidize it with the oxygen to make these quinones. And then the quinones can come together and they can polymerize. So I get a big long chain of these. I was looking for a better structure of this and I couldn't find it. So I'm just going to go with the big long chain of these. So I get a big long chain of these. And when you have, what happens when I have a lot of double bond, single bond, double bond, single bonds? Organic stuff, folks, or biochem folks. Resonance, good, resonance. What is that? How does resonance interact with light?
Conjugate.
They're conjugated, and this is something you guys definitely want to know because it's really important. This is why in organic, do you ever have like your reactions turn black? Maybe your reactions that turn black? Okay. Why do they turn black? Because you get a mess of something that is, it's not exactly this. But if I have a bunch of double bonds, single bond, double bond, single bonds, this gets really good at absorbing light. Okay, and the longer, so what you have is you have a difference, as you add those, you've got a, there's a highest occupied molecular orbital. Have you talked about HOMO and LUMO? Okay, and lowest unoccupied molecular orbital. What I have is, as I add more double bonds, those get closer together. If we talk about promoting an electron, sorry, Chem folks, if I got an electron here, right? I've got an electron here, and I want to go from here to here. As I add double bonds, they get closer together, and I need less energetic light to make that transition happen. Okay? And color is things absorbing light, things reflecting light. Okay? So if something's absorbed, you can get colors. So if you think about the red you see from tomatoes, that comes from lycopenes, which I don't know off the top of my head. They have like these things off the number something. But lycopenes are super long chains of double bond, single bond, double bond, single bond, double bond, single bond. Okay? So that's what's going on here. This is why this is brown, because these polymerize together. And I end up with a lot of double bond, single bond, double bond, single bond, double bond, single bond. Okay. All right, so why do the apples do that? Or the grapes, or the whatever fruit? What's the purpose of the apple for the tree? Why do trees make apples besides the fact that we cultivate? It's not sugar storage. That's a good idea, though. There was a lot of sugar in apples. What was that?
Reproduction.
Reproduction. How reproduction? What about reproduction?
I guess when apples fall, like, I don't know, the new seed.
Yeah, it's actually seed distribution, right? So you make out. So my parents have like a little apple orchard in their yard, and what happens is the deer always come out all summer and they're eating apples off those trees. Well, they eat the apples off those trees, which gets the seeds in their digestive system, and then they wander off someplace else and they poop out the seeds in a different location. They just moved the seeds for the tree from here to someplace miles away, and they deposit them with a little bit of fertilizer too. Okay? So the apples, at least originally, and the grapes and whatever fruit we're talking about, were about, the point that the plant makes them for is so that animals would eat these things, birds. So birds could be birds too, are going to eat these things. They're going to eat the seeds. They're not going to crunch up the seeds, or at least some of the seeds, and they're going to take them someplace else. Okay? So what happens if I'm an apple tree and I'm making apples and a bunch of bugs come in and start eating through the skin of my apples? They get through the skin of my apples and start eating my apples. Are they going to distribute the seeds for me? Flies aren't going to distribute my seeds. Fruit flies aren't. Let's take fruit flies. They're not going to distribute my seeds for me. These pigments are toxic. They're certainly toxic to insects, and they're toxic to bacteria that could get on the apple and try to eat down the apple once the skin's broken. They may be toxic to you too, but most of the stuff that's brown, there's not very much brown stuff compared to the rest of the apple. Okay, so it doesn't matter. You're big. You don't need to worry about it. Okay, but that's why plants do this sort of thing. Okay, so what Winemakers, let's say you're making a white wine. We'll get back to talking about brewing. Let's say you're making a white wine. How could you gotta mash up some grapes, right? How can you prevent this reaction from happening? Remove the oxygen. Okay. So that's you hear a lot about keeping oxygen away when you're making wine. And so when you've got all this mash around and stuff, you're not going to want to get tons of oxygen in there. Now, when you go to make the wine, let's see, I've erased it over here. I do want oxygen for a little bit because if I'm talking about sugar, what I need to do is I'm going to add some yeast, and I need my yeast to get going, right? I add some yeast, they need to make more yeast, and then they're going to run out of oxygen, and then they'll start making alcohol. Does that make sense? So when I put my bread yeast in here, this is like, it's probably a month old. Yeah, what's the date today? The 9th? 10th, that's almost a month old. It's about a month old, right around a month old. These things bubble like crazy when they start. And the first part is those bread yeast just growing and making more bread yeast and using oxygen. Okay? But once they start running out of oxygen, then they start making ethanol. They have to run out of oxygen. So you'll add some oxygen at some point. And winemakers will do what's called micro-oxidation. They'll add little bits of oxygen sometimes intentionally. Okay, to work on flavors. Let's see how hot that is. That's not bad. We'll let it go a little longer. Okay. So when you're making wine, another thing that they'll do, so you got to worry about oxygen. Okay. I think oxygen comes back later as well. One of the things that they'll do with wine as well is you'll take the wine, it will go to. You'll let it get through as far as you want. So let's say you're here and you wanted to have malolactic acid fermentation, and now you don't want the wine to continue to do this anymore. Okay, you're worried about somebody else coming in and doing other reactions, and I'll show you some other reactions. What they'll do is they will add stabilizers to wine that will stop the bacterial and yeast action. So let's say I want to stop it right here is where I found I've got the flavors that I want. What I can do is I can add to my wine metabisulfate, okay? And so metabisulfate has this formula, it has this structure. I left this on because I thought Bakerpedia was funny. And it will react with water to make sulfite. That's hydrogen sulfite, okay? Right there. So it'll make this, okay? And you hear about sulfites in wine, right? This is what they mean by that. And that sulfite can go on and it can make sulfur dioxide. And the sulfur dioxide, and the sulfite as well, the sulfur dioxide, it can help stabilize our wine. Okay. So, how does that happen? Well, the sulfur dioxide, it's a powerful reductant, so it can react with oxygen remember we're trying to get rid of oxygen? Okay, so if you have some residual oxygen around, the sulfur dioxide can actually react with that and pull it out of solution. Okay, our sulfur dioxide can become our sulfite again. And so many of the bacteria that we worry about in wine, or we don't want to grow in wine, need oxygen to grow. So you're going to slow them down by doing that. It also can diffuse through the cell membrane and reduce disulfide bonds. Okay, so it's going to affect our proteins. I'm going to talk about disulfide bonds in a second. But what I want to talk about first is it diffuses through the cell membrane. So what have you been taught about the cell membrane in biology? It's semi-permeable, right? It's a barrier to things going across, right? It controls what comes into and goes out of the cell. What sorts of things can cross the cell membrane, or sort of things don't cross the cell membrane without help? Very polar molecules, right? Okay. So our cell membrane is made up of these hydrophilic heads, and then they have hydrophobic tails on the inside, like this. And this is a barrier. Water doesn't want to get in here because it's polar. Sodium doesn't want to get in here because, well, it's not polar, but it's charged. Okay? So we have lots of different things that don't want to get into the interior of the membrane because this is very hydrophobic, so hydrophilic things aren't going in there. All right. However, sulfur dioxide is small. From my class in Gen Chem, you'll know it does have a dipole, but it doesn't have much of a dipole and it's not charged. And so it can actually get through the membrane really easily. Okay. So that's good for sulfur dioxide getting into the cell. A lot of pharmaceuticals, if you end up taking pharmaceutical chemistry here, what you'll learn is a lot of pharmaceuticals, they try to find things, when you try to make a pharmaceutical, you try to make it so that it is polar enough to dissolve in water enough, but so you can take it as a drug, but non-polar enough and small enough that it can get across the membrane on its own to some extent. Okay, so that's one of the things that you try to do. They worry about how making it non-polar, but not too non-polar. All right, so it's going to get across the membrane, and one of the things it does is sulfur dioxide does, it reduces disulfide bonds. So this is an example of protein. This is actually a trypsin inhibitor, so this inhibits digestion. But it has disulfide bonds that hold it in its shape. Okay, you see it as a very complex shape, and part of it staying in that shape are out of a bond here, and a bond here. Disulfide bond is a sulfur bonded to another sulfur across a bridge, and it's a covalent bond, so it's very strong in terms of protein interaction. Now here I found a paper where somebody was looking at how our disulfide actually, sulfur, what happened here? Sulfur SO2. So we got sulfur, how SO2 is involved in reducing, it's a powerful reductant. How we know it's a reductant? So let me go back to the structure here. Oops, wrong way. Okay, we've got these electrons here, okay? So it's a reductant, so it can reduce these disulfide bonds to open this up, and then my protein can unfold. And then here they were showing that then proteins, you can have a mess because individual proteins now have these open and they can refold. They did it at a high temperature. You wouldn't do it at a high temperature in wine. For those of you in organic, these are oxidized disulfide bonds, right? These are oxidized. This is reduced. This is not as clear that it's reduced. How do I know that this is reduced and that's oxidized? Folks in organic. Somebody in biochem.
More bonds to hydrogen.
More bonds to hydrogen, right. If you make more bonds to hydrogen, that's more reduced. Okay, so we think about that. Simple rule for that. More bonds to hydrogen is reduced, more bonds to oxygen is oxidized, and vice versa for both of those. So we could end up with a reduction of those sulfide bonds. So here's I said there'd be tangents, right? And you've already had tangents. But this also is why, if we think about when you get a perm, right? When you get a perm, this is going on in that as well. So your hair is a bunch of proteins that are kind of straight and long, that are kind of wound together. And what they do is they have disulfide bonds between the strands. So for folks who get a perm, what does it smell like? Maybe they've gotten better with this, but are they still stinky? Yeah, they're really stinky. Okay, so they're going to use a reductant, and things that reduce are generally stinky. So you're going to use a reductant to reduce the disulfide bonds in your hair. Then curl your hair into the shape that you want it in, and then you reoxidize, you remake those bonds in the new shape. And that's why a perm will hold your hair in terms of the curly shape. Okay. Now this actually is an overemphasis of this because you wouldn't do it to this extent here. This is what Nair does. These are still Nair. There, so the hair remover. If you take all those disulfide bonds and you reduce all of them, you break this up and the hair falls apart. So it's a milder treatment than that. Okay, let's talk about turning wine to vinegar. And this is a process that actually needs oxygen. So this is one of those places where you're adding the sulfur dioxide, and by adding the sulfur dioxide, you get rid of some of the oxygen. You're also inhibiting the bacterial growth because you're unfolding their proteins as well. But ethanol, you guys ever heard of alcohol dehydrogenase? So this bacteria can take, let's see, these are, I didn't put down, well, they're acetic acid bacteria, really, is what they're called. I forgot to write down the name here. But they take ethanol, and they can oxidize the ethanol. How do I know that ethanol has been oxidized there to make acetaldehyde? More bonds to oxygen, right? So I oxidize the ethanol. When I oxidize the ethanol, I actually I get to reduce, I get to take this thing called NAD plus and make NADH. This is something that's great because it's actually, you can use it to make ATP. So you capture a bunch of energy and you can use it to make ATP. And now we have acetaldehyde, and then we have aldehyde dehydrogenase, which then, is this an oxidation here going this way? Is that a yes or no? Yes. How do I know it's an oxidation? More bonds to oxygen again. And we oxidize it again. We can capture that energy that's released and make another NADH. Each of those NADHs is worth about 2.5 ATP. Okay. Yep. All right. So let's go back and try. Let's take a look at the supersaturation thing. All right. So supersaturation, concept you may have learned in Gen Chem. I haven't gotten that far in second term Gen Chem, know if we teach it or we skip over it. But it's possible to dissolve. So you talk about equilibrium, right? We talk about equilibrium and we have solubility constants. We haven't gotten to those in Gen Chem yet, but we will, okay? So you'll have constants and ways to calculate how much stuff you can put in solution stably. But it turns out you can actually put more into solution. You can put more of a solute into solution than can be stably held in there. And if you don't have any way to kind of knock it out or get it started coming out, it'll just kind of sit around. Okay, and that's called supersaturation. And you can do it for solids or gases. And some examples are, I've got sodium acetate and water here. I hope this demo is going to work, but I just put it together right before. We lost our preset demo, so I just put that together before class. Cloud seeding is an example of this. You've got a bunch of water in the air in the clouds. The air is super saturated with water, and if you give it a bunch of dust, essentially, you give it particles that the water molecules in the air can kind of stick on, and we call it nucleation. They stick on those particles of dust and it allows them to make a drop, then you can actually get rain, or you can get snow. You can have be super saturated with water and when it's cold out as well. So cloud seeding, when you hear about cloud seeding, that's what they're doing. They're just putting a bunch of kind of dust in the air. I'm trying to remember. There was some specific, when I was looking at this, I don't remember what the specific things they used for that. And then also you can end up with that for carbon dioxide in your brew. So I brought this month-old brew here, right? It is, I think it's bubbled a little bit, possibly because I agitated it. It's not absolutely done, but it's mostly done in terms of the production of carbon dioxide there. It's very, very slowly running along. I'll probably drink it next week, okay? Even though it's still bubbling a little bit. But you remember from your Gen Chem class, we have Henry's Law, right? The solubility of something in solution is equal to our Henry's constant times the pressure of that, the partial pressure of that solution, right? You guys remember this? Okay, and so the higher this partial pressure is, the more stuff goes into, the more soluble my stuff is in solution. Okay, so what this has to do with is, if I have my brew here, I'm going to put my brew in a, I guess we'll put it in a bottle like this. I have some carbon dioxide down here. Okay, and how much carbon dioxide in there is going to be related to the temperature. Okay, temperature, sure. But also what my pressure of carbon dioxide is up here. All right, so this thing has been sitting in my basement for a month. What do you think the pressure is of carbon dioxide right up in here? Any units you want. Two or three atmospheres. Too much, but thank you. Okay. 3 atmospheres is what's in a soda can. I got all sorts of trouble by saying that in my last lecture because you guys did the lab the next day, and it was. I told you the answer, I guess. But anyway, so at top of a soda can, that is goes. You hear it go, right? Okay. That is 2 or 3 atmospheres. Here, let's see. Everybody, be quiet. Hear anything? I didn't hear anything either. All right. So when I push it down, I got a little bit of stuff out there. But what I've got is, you see, I have this airlock up here. So it's actually pushing a little bit. But if this had 2 atmospheres on, this thing would be going bubble, 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 bubble out really fast. Right? It's about 1 atmosphere in here. It's maybe just a tiny bit more because this is displaced, but you're only displacing that much water. Okay, so not very much. So this should be, if I have 1 atmosphere of air up here, right? I should have however much carbon dioxide would work out if I used Henry's law. And so it shouldn't matter if I do anything to this. I shouldn't get more carbon dioxide coming out, right? Because if I agitate this, this is filled with carbon dioxide. I'm not going to put more pressure of carbon dioxide here because it's going to relieve the pressure here. Does that make sense? That this should be, if I don't have something like supersaturation. But if I do this, look what happens. Okay? What I'm doing there is I'm supersaturated. It's supersaturated, not supersaturated. It is supersaturated. And I'm stirring up particles. I'm getting it so the carbon dioxide in the wine runs into stuff, and it can form little particles, and it can make nucleation sites. It can start to make bubbles and make bigger bubbles, and the bubbles can come out. All right. You saw this actually in my last lecture if you were there, because what we did is we watched, it was Charles, and he had the big stirrer he put in. He got lots and lots of bubbles. I thought you might want to see it in person. But, you know, carbon dioxide is toxic to yeast, right? Just like it's toxic to us at certain concentrations. So if I really wanted to up the alcohol content in this, I could do this a bunch. I could have this in my office, and every time I come in, I could do that to it. I'm not adding oxygen because oxygen's out here. There's no oxygen in there by stirring it, right? This is all carbon dioxide up here. So oxygen's not going to come into it, so I don't need to worry about that. But I could essentially work on getting the carbon dioxide out of this thing, degas it, and then the yeast would feel a little bit better and they could make a little more alcohol. That's what I wanted to do. I'm too lazy for that, but that's one thing you could do. So let's go ahead and see if I can get this to work. So this is a solution of sodium acetate. So that's sodium acetate and water right here. It's a supersaturated solution. So in a little bit of water, I heated it up so all of it went in, and now it's cooled off, right? And so I don't know if it's cooled off enough, but if I go in and scratch it, I should be able to make a place for the sodium acetate to start coming out of solution, okay? Make a little deformity. So I'm going to see if I can get that to happen, or I'm hoping that, I can scrape a crystal down or something. And it may be this is just going to be a failure. We had beautiful setups for these, and I just couldn't find them before our lecture, so I made this. Yeah, it might still be too warm. Or it might be that I just need to, I don't have a crystal. Yeah, I got some sugar here. That's for my last one. All right, definitely I have particles in there now. Yeah, that's disappointing. Oh well. Okay. So I think I'll stop there. I've been going on for almost an hour. Does anybody have any questions? There is not going to be a tasting epic, but we did have a brewing club at one point. We could have a brewing club again, but students need to lead it. And if you're over 21, you can taste the stuff we make. Any other questions? Yes.
Sorry, the conjugates and stuff.
Yeah.
Are they all just like the aromatic ingredients, or does you had a ring and then it had two oxygen?
Right.
I haven't taken organic in a while, so I might be missing something. But does that bond break a long chain?
We have to look at it, but it does have to do with the protocol. I'm not sure what those bonds, whether you guys are oxygen or not. Like I said, I'm trying to find it. I was sure.
Oh, and now it's with coffee. I'm still.
But I was seeing bonds that those weren't oxygen. So we were getting it from the aromatic molecules. So you get around it somehow. I think it's around it. You get around it, and you're going to have one of the bonds in the ring goes to one electron and the next one takes the electron around and the next one.
Thank you.
You're welcome.
I was asking how quickly